I would like to thank Centro Congressi for this organization of this webinar. This is our annual webinar. Actually, it became a webinar. It was a seminar before, and we hope it will become again in-person seminar next year on DLB and on Lewy body disease more in general that we host in Chieti. I'm very happy to have world-renowned speakers today who will talk about the different aspects of LBD and biomarkers, and we'll keep updated on the new research that they are performing. So I will leave now the word to Professor Onofri, who will make a brief introduction, and then we can start. Thank you.
Okay. Thank you to the speakers for accepting to join us for this traditional seminar webinar organized every year by Laura. And I'm very glad to have here Professor Padovani, Doug Asland, Evelyn or Afina Lemstra, that I know her from some recent paper, which an editorial that she wrote, which intrigued me, but is far away from the topic that we'll be discussing today. I see Professor Doug Asland, who's present. I can see his video. So, well, I don't think I need to introduce Alessandro Padovani. He's the professor of neurology in Brescia and a long time involved with dementia, and actually on the run to be the chairperson of the Italian Society of Neurology. So he's talking about convergent and divergent pathophysiological pathways. In what? Alessandro?
Inizio io, no?
Sì.
Ok, perfetto. Credevo che ci fosse un'introduzione. I thought that Laura Bonanni was going to make an introduction, but anyway, ready. Now let me share our my slides, and I will be okay, ready. So, first of all, thank you, Marco. Thank you, Laura, for inviting me. It's always a pleasure to be here, even if there are a lot of things going on just in this period of the year. But anyway, I'm very happy to be here and to talk about convergent and divergent pathophysiological pathways. Now, I'm trying to put together what is coming up as an emerging evidence about the overlapping between what is amyloid-related disease and what are synuclein-related disease. In fact, both of them are multisystem neurotransmission diseases since the early stages. The evolution of dopaminergic and cholinergic dysfunction characterize both alpha-synuclein spectrum and the amyloid spectrum, and in both diseases, there is an early vulnerability of different neuroadrenergic and cholinergic systems. So I will go through this pathway in order to present you all the convergent evidence and eventually the divergent one. Usually, most of us classically tend to separate these entities in a very strict fashion. Alzheimer's disease is Alzheimer's disease, and this is different from frontotemporal dementia, and both of these diseases are different from DLB. But we know that Alzheimer's disease share clinical and neuropathological features with frontotemporal dementia and with dementia of Levy body. There are some specific neuropathologic features, but these are not as specific as we generally think. First of all, In the realm of Levy-Body disease, there is a strong overlapping of features between Parkinson's disease, Parkinson MCI, Parkinson dementia, and DLB. But all of them might present from the neuropathological point of view as a poor alpha-synuclein diseases, but in most cases, we have mixed Alzheimer's disease-related co-pathology. And this has been demonstrated by the extensive use of biomarkers and neuroimaging markers. And this is something that is clearly established. But when we turn in Alzheimer's disease, even in the realm of Alzheimer's disease, the pure cases of Alzheimer's disease probably represent no more than 10–25% of cases. In the large majority, there is a strong overlapping of features across all the spectrum of both Alzheimer's disease and other neurodegenerative disease. We cannot dismiss the fact that all of them, neuropathology associated with Alzheimer's disease or dementia, predict the, let's say, course of the diseases. And we have here a strong evidence that it's not only Alzheimer's disease related features, but also macroscopic infarcts, cerebral amyloid angiopathy, TDP-43, cortical Levy bodies, and eventually hippocampal sclerosis. In fact, when we talk and we look at prodromal PD-DLB, and we contrast this with prodromal Alzheimer's disease, well, there are a lot of clinical features and biological markers and even neuropathology that, as you can see here, make these diseases convergent to the same pathways or to parallel pathways. So alpha-synuclein, amyloid, tau, and eventual vascular pathology might characterize each of these classically separated diseases. Well, we know that amyloid accumulates in dementia Levy bodies and in Parkinson's disease dementia, but very few patients with Parkinson, truly Parkinson's disease, have amyloid accumulation. And as can you see here, the distribution of amyloid is quite different from Alzheimer's disease, but not that much. On the other sense, on the other side, we have the evidence that there is amyloid beta peptide deposition at the striatum of both Parkinson's disease with dementia and patients with dementia with Levy bodies. So it seems that in these different diseases, amyloid might play a role, maybe is an important role in terms of progression. In fact, there is evidence that the accumulation of beta amyloid might in fact be associated with a much more aggressive decline. When you talk about Alzheimer's disease, it has been already recognized for a long time that in the amygdala of a DEE patient, we might identify alteration very similar to those that we observe in truly poor Lewy body diseases. So there is a sort of, let's say, an orchestra of convergent pathway that might appear simultaneously in the same patient. Now, we might question whether we are facing a double trouble or on two different diseases. Are we talking about different subtypes of, and should we admit that Alzheimer's disease, as much as Lewy body disease, may present in a poor subtype or in a sort of overlapping subtype? Are these let's say, contemporary neuropathological feature, or really we have to admit the fact that some Alzheimer's disease patients have also synuclein alterations. Now, this is one of the most cited papers in the last year, and actually it shows that independent of early onset and late onset diseases in the realm of Alzheimer's disease, we have a huge amount of patients, up to, in this case, 49% in early onset and almost 40% in late onset Alzheimer's disease, the presence of Lewy body disease, in addition to TDP-43, in addition to hippocampal sclerosis, arteriophilic brain diseases, and vascular lesion. This seems to support the view that Alzheimer's disease is not as classically described as a single unitary disease, but we have different subtypes and different profiles. Well, there is no way that Lewy body disease are associated with synucleinopathies. And there is plenty of evidence that by using ultrasensitive RT-QuIC, we might, in fact, depict a sort of a cluster of diseases that share accumulation of synuclein. And as you see here, very few patients with Alzheimer's disease, PSP, and surprisingly, MCA might show an increase of synuclein. But when we look in this more carefully in these slides, in these papers, we may end up with some funny evidence. Does it depend on the selection? Are we biased? Because when we stratify patients according to CSF biomarkers, there is surprisingly a funny evidence that seems to confirm the neuropathological increase of synuclein-related neuropathology in Alzheimer's disease. In fact, by looking at this slide, you might realize that when we use a reliable biomarker like the ratio p-tau-Abera 142, you may end up with showing, in fact, that those that are truly Alzheimer's disease might show also an increase in synuclein levels at the CSF. So synuclein is there, and it is closely related to the neurodegeneration or tau-related degeneration, even if there is a relationship with amyloid accumulation. So amyloid accumulation and tau accumulation seem to be related to synuclein accumulation. And this is very important. We have tried to go in this direction, me and Andrea Pilotto and others with Zanusso, and we actually show that RT-QuIC might be a useful biomarker to define alpha-synuclein DLB, but as you can see here, there are also some negative subjects, and these are DLB ATN negative, but there is an effect related to the ATN and the CSF. But when we turn our attention on Alzheimer's disease by RT-QuIC, we were able to identify almost a huge amount of patients with synuclein levels, pathological levels. So these are Alzheimer's disease verified and certified by the CSF that shows synuclein alteration. Are these different from those that are synuclein negative? This is something that we will see in the next years. But this is what happened to see also in this already classical paper that Ian McKeith published by using DAT-scan. And actually, it was at that time that we realized that in possible Alzheimer's disease patients, there was a certain amount of patients that actually turned to be positive in the DAT-scan. And we discussed for a long time whether these Alzheimer's disease patient belong to the Alzheimer spectrum or to the DLB spectrum, or whether they presented contemporarily the two diseases. In fact, as you can see here, it is clear that the DAS scan is positive in patients with dementia, but as you can see here, there is a visible difference in individual with Alzheimer's disease compared to individual control patients or control subject. Now, there is a long story about the dopaminergic loss in Alzheimer's disease, and there is experimental evidence that there is a loss of dopaminergic hippocampal neurons in Alzheimer's disease mouse model, in particular in the ventral tegmental area, but not in the substantia nigra. And this is something important because this was reported by the authors and has been also by the same authors reported that the stimulation of dopaminergic system by the administration of the precursor levodopa-like, for example, also with selegiline, rescued the hippocampal synaptic plasticity and the dendritic spine density. And actually, this is still an open question. Should we treat those Alzheimer's disease patients with levodopa precursor or with, for example, anti-MAO inhibitors? Now, Venery made this also clear that there is a sort of relationship between the ventral tegmental area size and hippocampal size and memory competence. And this was also evident in healthy adults. And she made the claim that dopaminergic activity in the ventral tegmental area may be crucial for the earliest pathological features of Alzheimer's disease, suggesting new strategy for the early treatment. Now, we have evidence from this experimental study by Giacomo Koch that rotigotine might ameliorate LTP-like cortical plasticity in those patients with Alzheimer's disease that have reduced plasticity. In fact, rotigotine, not surprisingly, increased and normalized this pattern, as you can see here. And this is a sort of an indirect supporting confirmation about the hypothesis that dopaminergic might play a role. Now, as you can see here, as you can probably remember, we tried to use TMS in order to study the neurotransmitter features of both Alzheimer's disease, frontotemporal dementia, and Lewy body. And we concentrated our attention on those biomarkers like Psi, ICF, and CICI, because these parameters are sensitive to the dysfunction of cholinergic function, glutamatergic function, and GABAergic function. As you can see here, in fact, Alzheimer's disease and Lewy body share a cholinergic dysfunction. The difference that Lewy body disease show in addition to the corticobasal syndrome and PSP, also a dramatic decrease in GABA glutamatergic parameters, while Alzheimer's disease seems to be, at least in the early stages of the disease, in some way normal or not very different from controls. Now, when we put CGICF and Psi together, we were able, as you can see here, to separate Alzheimer's disease patient and DLB from PSP and control. So there are some overlapping features, the cholinergic dysfunction, and some divergent features, the glutamatergic GABAergic system. But DLB share glutamatergic and GABAergic dysfunction with frontotemporal dementia. Again, there is some convergent features with frontotemporal and divergent as well. So we are starting to look at these diseases as less separated, less divided or subdivided as we classically believed. Now I present some very, very, let's say, ultimate or recent data that with Andrea Pilotto started to collect by using DAT scan and looking not only on the striatal or basal ganglia that are characteristically altered in PD and DLB, but also on extrastriatal regions. And as you can see here, in fact, DLB might show a, let's say, a almost typical involvement of the cingulate, the insular regions, and eventually the thalamus. So remember, thalamus, insula, and cingulate cortex seems to be particularly affected in DLB compared to truly Parkinson's disease. Now, when we turn our attention to Alzheimer's disease, this is more or less the same. When we look at the impact of thalamus, insula, and cingulate, we were able more recently to identify a closer relationship with NFL, which is an indirect measure of severity of the disease. NFL predicts a sort of the clinical decline. And in fact, there was a strong correlation between the activity, the binding in the thalamus and the insula and cingulate with the plasma NFL levels. And this is quite interesting because in this way, we might use NFL and the DAT scan looking extrastriatal region as a predictive biomarkers for subjects with Parkinsonian diseases. But turning our attention to Alzheimer's disease, so remember, we have thalamus, insula, and cingulate. Now, looking at Alzheimer's disease, we were able to find, even in Alzheimer's disease, a sort of reduction of binding in the left and the right caudatus, not at the level of Lewy body disease, but still confirming what we observe in those papers with Jan McKeith. But looking at extrastriatal regions, as you can see here, again, cingulate, insulate, and thalamus, plus the midbrain, by confirming that probably the ventral tegmental area is affected in Alzheimer's disease. And with the extrastriatal binding and the DAT scan, we are able to see this. So again, divergent pathologies, divergent origin, divergent mechanism, but convergent pathways, not only from the neuropathological point of view, but also from the neurotransmitter point of view. And look at here, from the neuroanatomic involvement of the same region. So Alzheimer's disease is affected, and the dopaminergic pathway and probably the serotoninergic pathway as well, in the same region that we see or we have demonstrated affected in Lewy body disease. So I'm going to the conclusion. I think that Lewy body related and amyloid related disease are quite heterogeneous and neuropathological entities. While DLB PD is associated to specific synuclein related neurodegenerative mechanism, these are also associated to amyloid and tau, and both of these neuropathological features seem to play a significant additional role. Alzheimer's disease is also associated with specific amyloid and tau related mechanism, even if synuclein seem to play an additional, still unclear, but is becoming clearer and clear role. The use of a combination of biomarkers in the neurodegenerative spectrum can be expected to enhance our clinical diagnostic accuracy, especially early in the course of the disease, to characterize the stages, and probably, I'm very positive on this, accelerate the development of precise and effective disease modifying and neuroprotective agents. So thank you for your attention and for the invitation.
Thank you very much, Professor Padovani. Great lecture. I do have questions, but first there is a question by Camila Gonzalez. Hi, Camila. Is in the chat room. I'll read it for you. Do you think that terms such as pure ADs or pure DLBs are going to disappear and be replaced with the type of pathology present or not in this clinical diagnosis?
No, I think that Alzheimer's disease in the pure form is an entity, and that Lewy body disease in the pure form, it is an entity. What I'm implying is that we have to characterize all these convergent pathways in order to anticipate effects of those drugs when these drugs are supposed to manifest a disease-modifying effect. So I would use anti-amyloid in those patients that have amyloid, but I'm not expecting a strong effect in those Alzheimer's disease patients that also share synuclein alteration. And I'm expecting that in the future, we might use different drugs tackling different mechanisms in order to be truly effective.
Thank you. There is another question by Claudio Babiloni.
Ciao, Claudio.
Thanks to Professor Padovani for the very clear coupled alpha-synuclein amyloid neuropathology in a subgroup of AD patients. Can be this evidence inflated by either a misdiagnosis of Alzheimer's disease or mere comorbidity rather than causal interaction of those neuropathologies? Are there facts of topographic PET colocalization of amyloid and alpha-synuclein accumulation in that subgroup of AD with true diagnosis?
Well, Claudio, this is an important question. And is this a sort of a causal concomitant disease, or is there a true neurobiological interaction? I would prefer the last hypothesis because there is some experimental evidence that there is a let's say, cross peptide seeding. So a fibrillary amyloid might a sort of effect also to the synuclein aggregation, and synuclein aggregation might impact on amyloid aggregation. So these two mechanisms that we generally think are separated, in fact, they are not, and probably in some region they interact more closely because of the specificity of some neurons compared to others. So I think this is a new story that we have to explore because I think that in some way it is fascinating to strongly believe that in fact we we try to classify separate things, but in fact these things are more interactive than we generally think. So, but the implication is we have now the instruments to follow each single patient with different biomarkers along through all his course. And we have to start collecting data from different sources and follow this course in terms of biological biomarkers. At the end of this, let's say, enterprise, we might answer to the question.
Thank you. I do have a question, Alessandro, and I'm very curious about the GABAergic glutamatergic involvement in DLB as compared to other diseases. And as you know, we performed together in an Italian group of FTD last year a study on hyperconnectivity in prodromal frontotemporal dementia and Alzheimer's disease as compared to healthy controls, and we found an increased connectivity with EEG study in the very early phases of the disease. So my question is, can be that maybe the involvement of different neurotransmitter pathways is different along the line of the timeline of the diseases? Maybe we will have like a more early involvement of GABAergic glutamatergic system with hyperconnectivity, and then an involvement of cholinergic system in these patients. Maybe a stratification of the patients based on disease course could maybe answer the question.
Well, you're right, but we have to accept the limit that each of these techniques intrinsically have. We are looking at the brain and to circuits by let's say, by looking from a far position. So we are not there inside. But you're right. I think there are two forces that are working. The neurons are reacting to whatever the noxa is, and is reacting in two ways. On one side is trying to reinforce as much as it can his synapses and his connectivity, and actually it shows some sprouting of new synaptic activity that probably is not enough, is not, let's say, efficacious in maintaining the functional role of the neurons, and other neurons exhaust their activity. So you always see a sort of a positive reaction and a negative reactions. And what you picture, what you can evidence is let's say, a mean of these forces. So I'm not sure, but I didn't present some recent data that with Daniela Perani and Andrea Pilotti are collecting in Alzheimer's disease and in Lewy body. There is a stage where we see a loss of neurons, but an increase of connectivity. Now, and this is sort of so surprisingly, but it's so magic, it's so wonderful to see how can we, in fact, picture neurons that are losing their, let's say, their arborization, the axons, but at the same time try desperately to defend themselves and defend their specificity in terms of synaptic activity. Now, I think this is a new story, and I hope that you and Claudio Babiloni will be able to give the answer. I can give you some biomarkers, but I don't think I can be able to give you the answers.
Thank you. Professor Onofri, do you have any question?
Yes, well, actually, I was curious about the clinic. If, well, Alessandro's ever found a correlation between the clinical phenotype and the pattern of degeneration. I'm wondering, let's say, if you have patients where ventral tegmental area is involved, are these patients who have more hallucination inside their Alzheimer's disease, or patients with DLB who have low GABA levels in the insula? Do they have more somatoform disorder than others?
Well, the only feature we were able to evidence so far by following the patient for 2 and 3 years, those Alzheimer's disease with synuclein, CSF synuclein, and those DLB with, let's say, amyloid evidence or dopaminergic dysfunction. Well, at the baseline, they're not different at all. And the only thing we see is that there is a sort of a more rapid decline, claiming that to have 2 diseases is even worse than having just one. But this is a very simply answer. We will follow the patient in the next years, and try to detect some differences along the spectrum. But so far we didn't find any evidence of a strong difference.
But, well, actually, let's play again around the Occam razor and just never considering 2 diseases as coincidence. So
you're right.
But the point is that really the need to understand how these mechanisms are impinging on the development of the phenotype is, I think it is also for the development of treatment is a radical cue. Can I comment on one smaller thing about hyperconnectivity? In the paper that Laura wrote around hyperconnectivity, we also suggested an alternative hypothesis. And the hypothesis is linked to the pathway of apoptosis in each cell. When a cell is dying, the membrane potential shifts, upwards. So that's making the firing of synapses quite more effective, quite not effective, malfunctioning. So it is a malfunction rather than a compensation. And my question was, why do you have symptoms if it was a compensation? So I believe it is more of a mechanism of apoptosis.
Oh, nice point. I think that when we see the patient, the system is already, let's say, on the side of having exhausted all the, let's say, resilience mechanisms. So the system in some way is losing the efficacy, while there are spots or parts of the population that are still trying to maintain the resiliency. So I wouldn't be as much as you are sure about the fact that there is no compensatory effect. There must be some compensatory effect because let's say the asymptomatic stage is pretty long. And even in patients with at the beginning of the disease, let's say in the MCI stage or the prodromal, whatever it is, there are some patients that actually has a very, very slow decline. So I think probably there are different mechanisms explaining the hyperconnectivity, but to me it makes sense that the neurons that are there have some defensive mechanism, and the defensive mechanism in neurons is to maintain the connections, and to maintain the connection when you are losing some connection is to replace the connection to maintain the system efficient. So if I have a company and I have 100 people working with me and I lose 20 people, I have to ask the other people to work more. And those will eventually get a burnout syndrome if they work double or triple. So at the end, I will lose the company anyway. But in the meantime, I will compensate for the losing of the 20 workers. So I think this is a sort of a very simplistic reasoning, but I think the system can accomplish some loss, but at a certain point, it will have to pay, the efforts to compensate.
And lots of room for more research. I think right. Laura must introduce now Doug Arsland as an interview.
Yes.
Okay.
So thank you, Alessandro, for this beautiful talk. We hope that you can stay longer with us.
I will try.
The discussion. And I'm very pleased to welcome Professor Doug Arsland, who is the chair, the coordinator of the European DLB Consortium, and former chair of the ISTAR Professional Interest Area on LVT. And it's a real pleasure to have him here. He's a friend and very happy. His talk will be on cognitive and behavioral aspects on Lewy body disease. Thank you, Doug.
No, can you hear me now?
Yes.
So thank you, Laura, for the kind introduction, and thank you for inviting me. It's a pleasure to be in Italy, even virtually. So many great collaborators and friends, beautiful country. So it's a shame we have to do it virtually, but we can look forward to a real meeting soon. So I assume I have more. I have my 20 minutes, Laura. We are a little bit late, but I will talk then about cognitive and behavioral aspects of Lewy body disease, presenting some work of my many fantastic colleagues. Some of them are joining this meeting. Now I was struggling to move my slides. Here we go. So I was planning to start a little bit with some pathology and circuits, but now Alessandro has explained everything so very nicely, so I don't need to spend much time on that, but just I always like to present Glenda Halliday's lovely cartoon showing the association between Lewy bodies and tau and amyloid pathology and the sequencing of dementia and motor symptoms in Parkinson's disease and DLB, and it really clearly shows the continuum of this Lewy body disease. We have on one end, we have Parkinson's patients who can remain without dementia for a long time. On the other end of the spectrum, we have DLB who start with dementia and then Parkinsonism develops later, and they both have Lewy bodies, but more in the cortex in DLB, and DLBs also have more amyloid pathology, as we heard from Alessandro. But then there is a large group in between there, Parkinson patients who develop dementia fairly early in disease. And one of my main research areas has been, how can we predict, how can we understand why some Lewy body disease patients develop dementia early and others late? And I get back to the clinical ways of assessing this. So this will be a very clinically focused presentation, but I will also highlight, again, nicely and elegantly shown by Alessandro, but just remind ourselves that although Parkinson's disease is seen as a motor disorder, a nigrostriatal dopaminergic disease, as we have heard, it's actually a multi-system disorder with involvement of not only the brainstem, but the forebrain, the limbic structures, and the neocortex in the majority of patients, and that a number of neurotransmitters are involved in addition to dopamine, as we also heard from Alessandro. So with this, there's no wonder that there is a clinical spectrum in these diseases, which is complex and involves not only motor, but also cognitive and behavioral changes, in addition to autonomous and others. So I will focus on cognition and behavior. So this, I think we have to show McKeith's criteria paper. What is DLB? What are the core features of DLB? In addition to having dementia, which is a core, which is the basic for dementia with Lewy bodies, we have the four core features: fluctuating cognition and hallucinations, recurrent visual hallucinations. So just to demonstrate that the behavioral or the psychiatric features of the disease in DLB are actually part of the definition. So clearly, this is an inherent part of the disease. This is not something that comes in addition to it. It is a very central and very fascinating and also important part of the disease from a clinical point of view. So as you will have heard, many of you, there's increasing focus on the early stages, not only of early stages or prodromes of Parkinson, but also the prodromal stages of DLB. And we have this paper published in 2020, which presents the research criteria for prodromal DLB, and many of the faculty and speakers on this symposium are on this on the author list. So this paper in particular focus on the MCI stage of Lewy bodies, of DLB. So some DLB patients, probably the majority of them, start with a cognitive syndrome and MCI, which is often non-amnestic, but can also be amnestic. So MCI, and then addition of one or more core features, hallucinations or Parkinsonism. That's the criteria for MCI Lewy body. In addition, this paper also suggests three other types of prodromal, which includes psychiatric symptom in older people moving over to DLB, or delirium type, or REM sleep behavioral disorder. And we know a lot about the latter one, and we know quite a lot about MCI, but we know very little about the psychiatric and the delirium type of prodromal DLB, and that's currently a very important area of research. I just wanted to mention this paper recently published, led by Alberto Jaramillo Jimenez, who is on, I think, in the audience, and also with Laura, a patient with our colleagues in China who had as her first symptom panic attacks, which occurred late in life, and then gradually developed a full-blown DLB. which was also verified by dopamine transporter SPECT and EEG. So this is one of several case reports showing that psychiatric symptoms can actually be the first symptom of a DLB syndrome. I also want to just highlight again the frequency and importance of psychiatric symptoms in Parkinson's disease. So these are two cohort studies, one old Norwegian study from Stavanger and the PPMI study by Dan Weintraub to the right. And we can see here that a number of different psychiatric symptoms are very common also in Parkinson's disease, not only in DLB, but also in Parkinson's disease. We can see depression, anxiety, hallucination, delusion, apathy being quite common. And interestingly, even at the time of diagnosis, we might see some of these psychiatric symptoms. So these symptoms are part of the disease, also in Parkinson's disease. We heard the potential mechanisms earlier, and they play a large role for the patient's quality of life, for the health economy consequences, and also for the care. So it's very important for clinicians to be aware and look for these symptoms also in Parkinson's disease. And similarly, this is data from the Norwegian DEM West study showing the frequency of a variety of psychiatric symptoms also in DLB and at time of diagnosis. And we can see the red bars of the percentages in DLB of the MPI psychiatric symptoms, and the blue bars is Alzheimer's disease. So we can see that although psychiatric symptoms are common in Alzheimer's, they are more common in DLB, as you would expect, given the mechanisms that we have heard earlier today. So psychiatric symptoms are common in DLB, but also in Parkinson's disease. So then the most specific and fascinating symptom of both Parkinson and DLB is the psychosis or the visual hallucination, which is a typical psychotic symptom in both disorders. And this is work by Dominique Fitch, who's my good colleague and neighbor here at King's, and illustrating a typical sequence of symptoms. Many, maybe not all, but most patients start with very vague, mild symptoms like illusions of false sense of presence, which then develop to more hallucinatory experiences with well-formed images of animals or people. And sometimes, not always associated with delusions. Delusional content can be delusions of jealousy or of theft. And these symptoms are then often related to more cognitive impairment, although they can occur also in cognitively healthy people, but the level of insight, which is very important, varies with cognitive impairment. So people with dementia often lack insight, so they act on these symptoms, they think they are real, which can cause problems for the patient and for the carer, whereas with cognitive intact patients, often, but not always, often have insight that these are disease phenomena. Cognitive impairment is essential in DLB. It's essential for diagnosis, but as we heard, dementia does not need to be part of the disease. It occurs also at the pre-dementia stage. And I want to just stress that although memory is common, the typical scenario is more that the attention executive and visual perceptual disturbance in these patients. I'm running a little bit rushing so that we can have Evelyn's talk also in time. I want to also highlight the cognitive element in Parkinson's that I mentioned earlier. This is, to the left, you see a graph of more than 200 patients, again from Stavanger, an early cross-sectional study, epidemiological. It's a community-based study, and we can see that the MMS score range from the normals down to 0. So we have the full spectrum of cognitive impairment, not only mild impairment, but even severe dementia. And on the upper right, you see the much cited Sydney study. We have similar data also from Stavanger showing that whereas per definition, Parkinson's patients do not have dementia at diagnosis, most of them, if not everyone, develop dementia if they survive. So after 10 years, approximately 50% of them will have dementia. So Parkinson's disease is a dementing disorder. And cross-sectionally, we see patients with normal cognition, we see people with mild cognitive impairment, and we see people with dementia. Roughly 25% of Parkinson patients will have dementia. So this is work from Lucia Patsu and Rai Chaudhary and our group recently published in Nature Neurology, a review of cognitive impairment in PD, where you can find more details. But it illustrates the progression of disease, but it also illustrates the variation here, this curve here. So it's not a gradual progression. Some people develop MCI, then they go back to normal, and they go back to MCI, and then progress to dementia. We can also see that there's there's a big difference in the rate of cognitive decline, and also the time to develop dementia, which is again an important factor to consider. So I think maybe in this distinguished company, I think we just skipped the assessment of cognition. Just want to highlight that MoCA is an excellent screening tool, short, and also gives us some cognitive profile information, which is relevant. I want to highlight also the Diamond Levy study program, which has a really excellent description of how to diagnose both the cognitive and psychiatric and other aspects of DLB and Parkinson's disease. So skipping that and moving a little bit to prediction of cognitive impairment. This is early work from both Stavanger and the Pittsburgh group, showing that a beta pathology which can be measured in CSF is a predictor of future cognitive decline in Parkinson's disease. So that's one way of predicting who will develop cognitive impairment early on, and many patients are asking for that. Yes, I have Parkinson's disease, but will I have dementia? And this is one way of predicting this. So then I want to present a new study, which was first authored by Carmela Gonzalez, who we already seen in the audience. With the EDLB study, many participants in the audience here, and we can see that they look for plasma markers of p-tau 181 and 231, analyzed by Nick Ashton and Henrik Zetterberg in Gothenburg. And we can see DLB patients had somewhat increased levels compared to controls, but lower than Alzheimer's disease. But more relevant here is that having a high level of p-tau 181 and 231 was actually a predictor of more rapid cognitive decline. Not a very strong effect, but again, demonstrating that if you have dual pathology, that's a risk factor for more rapid cognitive decline and earlier dementia and Parkinson, rapid cognitive decline also in DLB. So we've done a similar study, not yet published, in Parkinson's disease based on the King's cohort, or international NILS cohort led by Rai Chaudhary, where we looked into whether these plasma markers could predict cognition. We, unlike the DLB, we found that p-tau did not predict cognitive decline, but NFL did. So as we heard earlier from Alessandro, NFL is a new and really interesting new marker, and having high levels of NFL in Parkinson was a predictor of more rapid cognitive decline. So this is a better way of predicting cognition, better than CSF, less invasive and cheaper also. So I want to end with last 5 minutes on treatment. So what do we know about drug treatment of cognition and behavioral symptoms in DLB and Parkinson? Well, we know regarding cognition that the cholinesterase inhibitors, particularly rivastigmine and donepezil, there's good evidence that it can have a positive effect on cognition in both DLB and in Parkinson's disease with dementia. So that should be the mainstay therapy. Obviously, we all know this is by no means a cure, so patients tend to continue the progression, and we don't yet have any preventive treatment, unfortunately. So what about hallucinations? There's quite good evidence in Parkinson's disease, mainly on clozapine. Some early studies with small studies was strong effect in showing that clozapine is probably the best drug for psychosis in Parkinson's disease, but used infrequently because of all the hassle with blood testing. We have new data with pimavanserin, which is now FHT2A inverse antagonist, which is approved by the FDA, but not yet in Europe. but there's good evidence that it has a good antipsychotic effect. In DLB, we know much less. We'll show the data on PD later on, but there are actually no good studies. We have some studies with aripiprazole indicating effect. Regarding the other studies, we really don't know, and there are lots of safety issues related to this. And with depression, there's good evidence in Parkinson's disease for antidepressants, but no evidence in DLB. I think maybe just skip this. We discussed it already. I just wanted to show before we end an interesting study of pimavanserin in dementia. This is in the term dementia-related psychosis. They recently published a Harmony study. This included mainly Alzheimer's patients, but a small group of DLB patients. So we don't have a specific DLB study yet, but this study showed that pimavanserin could prevent relapse in patients who had had psychosis, dementia and psychosis, who had a remission on pimavanserin and then were followed, and those on drug had less risk of relapse than those on placebo. So that's a very interesting and important study, and we need something similar for DLB. And then just to say before we end this, luckily a good activity ongoing and drugs in the pipeline. We have Antorin, which has a gut-brain axis acting drug. We have the PD-Mind, which is a PDMCI trial where Laura is involved, the nicotinic agonist. We have Arvid Rongve, who I think also is on the audience list, who is leading an Ambroxol study, the ANID in Norway. We have John Paul Taylor's Cobalt with combining memantine and rivastigmine that's just started. We have interesting new agents, including neflamapimod, both as a disease-modifying drug with P38 MAPK inhibitor. We have ondansetron, which is currently being tested by the UCL group for psychosis in Parkinson's. It's a 5-HT3 antagonist, and we have interesting studies ongoing here at King's with cannabidiol for psychosis in both Parkinson's disease and Alzheimer's disease. So hopefully, with a couple of years, we will have more data on these new exciting drugs. And then to summarize, we've seen that cognitive impairment and visual hallucinations are key cognitive and psychiatric symptoms in both DLB and PD, that clinicians need to be aware and then actively ask for these symptoms, and there are instruments available, that there are biomarkers that can predict the rate of cognitive decline in DLB and Parkinson's disease, and that drug treatments are available, but that more and better alternatives are urgently needed. So with that, I thank all my collaborators who have helped, Laura and others, and look forward to the rest of the symposium.
Thank you so much, Doug. Thank you. I would like to first reassure the audience that we won't have, unfortunately, the discussion after Roberto Ceravolo talk, which is pre-recorded because Roberto is not here today. So I can take the time to make a question to Doug, and I would like to ask if he thinks that maybe the first appearing symptom in DLB can be in some way a prediction of the subsequent trajectory of cognitive decline. For example, if we have like a fluctuator or more a patient with RBD or with Parkinsonism as the most important core feature, can we predict a different decline?
That's a very good question, Laura. I don't think there's evidence yet. I don't think there are any, let's say, long-term longitudinal studies of patients with prodromal DLB, particularly not with psychiatric onset, but I think it's possible to, and you gave me just a new idea of a new study. So in the Norwegian DEMVEST study, we included patients at time of dementia, but we asked retrospectively for what was the first symptom. So we know that, at least from with all the biases inherent in that, we know to some extent what was their first symptom, and we have, as you may know, these are patients that have been followed for until death, actually. So we know the full disease course of them. So that's actually a brilliant study, and when this seminar is over, we will start looking into that with data all there. So brilliant question.
Thank you. And there's a question by Claudio, who is always very active, and we thank him for this. So thanks to Professor Arzon for the overview on the DLB PD clinical features and treatment. What about the outlook of anti-amyloid drugs in DLB with amyloid neuropathology?
Ciao, Claudio, thank you. Brilliant question, and obviously that is something we have high hopes for. So one of the good things for working in DLB is that everything that is relevant as a therapy for Parkinson's or Alzheimer's disease is also relevant in DLB. So I fully agree with you that is an interesting study. We first have to show that the anti-amyloid drugs really work in Alzheimer's disease, and as you know, that's still under debate. Unfortunately, so Biogen now withdrew from the EMA approval, but then there are other drugs coming up. So Claudio, next time I'm in Rome or we meet in Newcastle, let's have a bottle of red wine and then discuss the design of that study. Obviously with EEG as a biomarker.
Thank you. Professor Onofri would like to ask a question or introduce the next speaker. It's not there. Okay. So thank you, Doug. Thank you very much for being here. So if Professor Onofri is not here, I take the way to introduce Afina Lemstra, Evelyn Lemstra, who works in Amsterdam, is a friend also. Very happy to have her here. And she's involved with us in the steering committee of a European DLB consortium and performs lots of studies on fluid biomarkers and also neurophysiology. She will talk today about the fluid biomarkers on LBD. Thank you, Evelyn.
Thank you. Thank you, Laura and Marco, for inviting me for this nice seminar or webinar. Let me see if I can share my screen. And I will focus on fluid biomarkers in dementia with Lewy bodies, which is, I think, a very interesting entity between Alzheimer's and Parkinson's disease. I have nothing to disclose for this specific meeting. And why are fluid biomarkers interesting? There are many interesting aspects about them, but it's if we look at neurodegeneration, and I think Professor Padovani already touched on this, none of these diseases are actually single protein diseases. There are many proteins involved in various ways. And the nice thing about fluid biomarkers is that we can actually measure direct disease processes instead of indirect measurements like imaging. There's also the possibility to measure multiple proteins at the same time, which gives us some tools to understand biomechanisms and also which kind of disease processes are ongoing in a single patient. Several studies have already, and most of this work is done in Alzheimer's disease, but have already shown that these disease processes are detectable in fluids in early and prodromal phases of disease. And fluid biomarkers have both diagnostic and prognostic properties. Compared to some more expensive biomarkers like PET imaging, most of these fluid biomarkers are easily obtained and relatively cheap. One of the main issues in bringing these fluid biomarkers to its full potential and use in routine clinical practice is standardization of assays and harmonization of cutoff values, etc. Let's see. Next slide. So I will talk today about fluid biomarkers in DLB, and although there have been many markers studied in experimental studies, I will talk about three important ones which are available or almost available for clinical practice: alpha-synuclein Alzheimer biomarkers and neurofilament light. And I will give you an update on these three biomarkers, and I will close with some talk about how can we use these in clinical practice. So as mentioned before, the pathology in DLB is, of course, it's a Lewy body disease consisting of alpha-synuclein aggregates in Lewy body and Lewy neurites. And we find this particular pathological process around, diffusely distributed around the brain. But a substantial portion, and if you look at post-mortem data, up to 80% almost, we find AD changes, Alzheimer pathology, consisting of amyloid plaques and tau tangles. So what about alpha-synuclein? This is the main culprit. Alpha-synuclein can be measured in several body fluids, in blood, in CSF, and also in saliva. And recently there has been more research also on peripheral tissue in which we can measure alpha-synuclein. And probably the pathological forms of alpha-synuclein are not, it's not the alpha-synuclein protein itself, but is the pathological oligomers and phosphorylated alpha-synuclein. What do we know by now? There has been a large meta-analysis which was published several years ago on total alpha-synuclein in Parkinson's disease, and there are fewer studies on dementia with Lewy bodies. And it has a lower, on a group level, we see that Parkinson's patients tend to have lower alpha-synuclein levels in their CSF. These are mostly CSF data, with a moderate sensitivity and even less specificity for Parkinson's disease versus controls. However, there is a lot of heterogeneity in the assays that were used in these studies. There are no standard cutoffs, and many of the studies were also biased, especially very highly selective patient populations. So this is not something that has been brought forward to clinical practice, and I hope that the efforts that now are undertaken to standardize the alpha-synuclein assays and also the oligomers will help us a bit forward in this. As I said, probably the more pathological associated proteins of alpha-synuclein are the oligomers and the phosphorylated alpha-synuclein. And we know from many studies that these are increased in Parkinson's disease. In dementia with Lewy bodies, the results are less consistent, and the results are hampered by small studies with not many patients involved. You see here in this figure a study that we carried out from our center together with Professor Alagnaf. And what you see here is that there is some elevation of or some lowering of total alpha-synuclein in DLB and PD as we would expect it. And there is some, on a group level, a significant difference between non-Lewy body disease and Lewy body diseases on the oligomeric and unfortunately not on the phosphorylated alpha-synuclein. There have been other studies that have shown some more convincing results, and maybe we also have to use ratios to make a discriminative features of this. But yeah, we need more studies for this particular biomarker. A very promising technique is what we call RT-QuIC, real-time quaking-induced conversion. It was first developed for prion disease, and it takes advantage of the capacity of proteins involved in neurodegeneration to self-propagate. And so just a small explanation on how this works, and this is a figure that was published by Inga Zer in a comment in Lancet Neurology, is that you have this misfolded protein, and in this case it's alpha-synuclein. And this is coming from a patient It can be a body fluid like CSF, or it can be maybe tissue in which the pathologic protein is found. And you combine that with a recombinant protein, and then you see this interaction. Then they like glue together, and due to this self-propagating properties, we see like a catalyst reaction, and you see spontaneous aggregation and conversion from the normal, let's say normal recombinant alpha-synuclein into pathological aggregates. And we can amplify this process by quaking, and you get a signal due to a fluorescent agent attached to it with a kind of thioflavin, and that you can use as a readout. And several studies, especially in Parkinson's disease, since this was also adapted for alpha-synuclein after very successful results in prion disease, show very high sensitivity and specificity for this pathological processes. One of the downsides is that it gives only qualitative outcomes, so it's either normal or abnormal, although there have been some attempts recently to try to make a kind of quantitative RT-QuIC assay. A great thing about this is that it was at first developed for CSF, but it has been adapted now with quite some good results for other more easily accessible or easily obtained tissue like skin and saliva and olfactory mucosa. One of the questions for alpha-synucleinopathies is that we know that different strains of alpha-synuclein are involved in alpha-synucleinopathies, and RT-QuIC, well, we don't know if it can differentiate between these different strains. And again, we need more standardization of assays before we can use this in clinical practice. What do we know about RT-QuIC in DLB? Numbers have been not that high in the studies that have been performed. This is a very, I think, readable and very clear review on RT-QuIC in general by Alison Krauss, recently published. And I realized that these are very small letter types, but what I want to highlight is that there have been several studies, and I have to say that the Italians are really leading this field, where we see small groups of DLB patients included, usually in comparison with other synucleinopathies. And I think the bottom line is that it shows very high sensitivity and specificity. So it looks like a very valuable diagnostic test, and it has been already studied in various tissues. Another interesting thing of RT-QuIC is that it can also detect pathological alpha-synuclein in early stages of the disease. As you are probably all aware, Ramsay behavioral disorder is one of the most remarkable syndromes associated with alpha-synucleinopathies. And in a recent paper published by the Spanish group of Alex Iranzo, who looked at 52 Ramsay behavioral disorder patients compared to 40 healthy controls with a considerable follow-up. And he looked at CSF alpha-synuclein with the RT-QuIC assay, and patients who were converting to Parkinson's disease or Lewy body dementia in the RBD group had a considerable number, had a positive RT-QuIC test at time of diagnosis of RBD, idiopathic RBD, this still without the other symptoms of an alpha-synucleinopathy. So this is very promising, I think, to use this test as a prodromal marker, especially if we manage to do this in easily obtainable tissue or fluid like skin or olfactory mucosa, especially in this Corona era. Everybody's used to getting a stick up his nose, so it shouldn't be too much of a problem. although there has been another study on that already published, in which the sensitivity was less remarkable as the high specificity we see in CSF. We have worked together with Piero Barci, and then we worked on MCI patients, and we compared 81 MCI patients with Lewy bodies and Alzheimer's disease with controls. And also in this group of patients, we find a very high diagnostic accuracy of RT-QuIC to discriminate Lewy body disease from Alzheimer's disease. Of course, and it has been already mentioned before, I think it would be very interesting to look at the performance of RT-QuIC in other prodromal Lewy body cases, like late onset psychiatric cases and delirium, but these studies, as far as I know, are not being undertaken yet. So just to conclude, I think RT-QuIC is very promising, and it's not available, I think, in most countries for clinical practice, unlike the prion disease, but I think it, once we get the standardizations right, and there is a commercial RT-QuIC assay already approved in the United States by the FDA, so I think it's coming up soon to be used in clinical practice. So what about AD biomarkers? We can measure AD pathology with a very high correlation with the actual brain pathology, as we know from the Alzheimer's studies that have been performed extensively, and we can measure amyloid beta 42 tau and phosphorylated tau in both CSF and blood. The European DLB consortium which is a great consortium doing a lot of research in DLB. One of the first things we did was look at CSF ADB markers across alpha-synucleinopathies, and there we showed that a very high percentage of DLB patients has amyloid, decreased amyloid in the CSF, and to a lesser extent also phospho-tau. It's not comparable to the high prevalence in post-mortem studies, which is up to 80%. So maybe, well, one could think of that it's AD pathology is evolving during the disease process, but that's also, we have no longitudinal data yet, working on that. And we and others have shown that AD pathology is not just like some bystanding process. It has actually clinical impact. It's causing more severe memory impairment in DLB patients. Also, in our study, we showed more hallucinations that has not been reproduced across all these cohorts that has been studied. More tau was in another cohort related to less Parkinsonism that also has to be reproduced. And we see more hippocampal atrophy in DLB patients who have AD pathology based on CSF biomarkers. It also impacts disease progression, and this is data of a recently published study from our group where we looked at over 200 DLB patients with CSF, and we used the ATN classification, as you are probably all familiar with. And we see that in this cohort, almost 60% of the patients fall into the AD continuum. And if we look at cognitive decline based on MMSE data, we see that patients within this continuum have more steep decline over time. And this is also true for survival. Patients with AD pathology, they will die sooner than patients without AD pathology. So it's not, it has very relevant clinical impact, and as already been said before, I agree these patients should be included in trials with amyloid lowering agents. Going to blood-based AD biomarkers, I think that's a very interesting development because it's much easier to obtainable, and we can do much more studies on AD pathology in various neurodegenerative diseases. It has already been extensively studied in AD. And we know that measuring A beta as a plasma biomarker has some downsides, and most importantly, it has less contrast with non-pathological A beta, which is produced in blood cells. So for diagnostic purposes, it seems more logic to use A beta 40-40 ratios to account for that. And much more promising even is, I think, the phosphorylated tau, in which you have various species which correlates to AD pathology in the brain. What do we know on dementia with Lewy bodies? Well, Dag was ahead of me with showing this beautiful paper that was co-authored by many of us, but I think Camilla did a great job writing this up for JAMA. And what we see is that there are a considerable number of patients with p-tau, as we see, not as high as AD, but higher than healthy controls and PD, and it has it looks to be correlated with a decline in cognition. We cannot only measure p-tau, but we can also look at other blood-based biomarkers, and many are being tested and researched at this moment, thanks also to the development of very high-end immunoassays that we have at this point, at which we can measure several proteins at the same time. This is a paper recently published by the UK group, and they look at different biomarkers. They look at not only at p-tau but also at the amyloid markers, neurofilament light, and GFAP, which is also a very interesting marker. We know that it's elevated in Alzheimer's disease, and it's a marker for inflammation. And in this study of considerable size, we see that it's not only elevated in AD, but also in Lewy body diseases. We have been studying this in the EDOB cases that were also used for the study I just mentioned, which was published on the p-tau in JAMA Neurology. We're now looking with this same 4-plex immunoassay, the Simoa, at the EDOB cases, and preliminary results are that there is also a negative correlation with baseline MMSE, and that we see higher levels of GFP and NFL associated with Parkinsonism, but not with other core features of DOB. We're still working on this, and we're also hoping to include some longitudinal data. Then the third biomarker I want to talk to you about is neurofilament light. It has been mentioned before, but it's a very interesting biomarker, as we can measure it both in CSF and plasma, and they have high correlation. We know that neurofilament light elevation is associated with axonal injury, so it's a kind of measure for neurodegeneration. It's not very disease specific, and there is an age dependency, so you have to correct for age if you measure neurofilament light. And here you see a few, the Lewy body disease highlighted, but I will zoom into that a bit. This was a recent paper also from the Parche group, in which they show that neurofilament light is not really elevated in PD, but is in atypical Parkinsonism. And this is a large meta-analysis review by Bardell from our center here in Amsterdam. And she showed, here you see the healthy controls, you see the age-dependent concentration levels of neurofilament light. And this is the line for DLB. There is some elevation, but not as high, for instance, as FTD or HIV. And in Parkinsonian disorders, you see that for Parkinson's disease, there is not really a change compared to control. So it's maybe it has some added value in on a group level to discriminate atypical Parkinsonism for Parkinson's disease. And also it could have maybe some prognostic value. This was a small study published by Pilotto and then from Professor Padovani's group. And there they show that on a longitudinal decline that neurofilament light has an impact. And I think this is a very interesting study, and that it should be reproduced in larger cohorts. To conclude, because I'm running out of time, I think we have some great biomarkers in place and ready to be maybe implemented in clinical practice. We don't have really a diagnostic biomarker yet in our clinical criteria, but ARTIQ I think it's very promising. And once we get our standardizations right, and we can show that ARTIQ, because what we need is study in a large real life cohorts, then ARTIQ can be very promising to apply for diagnostic purposes. I think the Alzheimer biomarkers we can use not so much for diagnosis, but for prognosis and also for stratification for trials. And then of course, blood-based biomarkers are much easier to obtain than CSF. And maybe in future there we can use biomarker panels, including also NFL and maybe GFAP or other markers that are specific for certain disease processes to stratify patients and to maybe also learn something a bit more about several disease processes within a singular patient and its prognosis. With it, I want to conclude, and I want to thank everybody for organizing this and for your attention. I'm happy to answer some questions.
Thank you so much, Evelyn. We have a question from Claudio. Thanks to Professor Lenstra for this overview. We wouldn't expect that, right? The Claudio on the innovative fluid alpha-synuclein biomarkers of LVD versus AD, how sex resilience factors, cognitive reserve and lifestyle, or risk factors, hyperglycemia, neuroinflammation, can interact with the diagnostic and prognostic value of these fluid biomarkers?
Yeah, that's, I think, a very good question. And as I said, I think we're still in the process of looking at all these details. There's a lot of biomarker studies done in selected patient populations, and I think the impact of environmental factors and other factors are something we still have to research. And I think also genetic factors are something that is very interesting to look at. Yeah.
Thank you. There's another one in the chat room. Doug, very nice talk. Doug, you can make the question online, live.
Oh, okay. Well, thanks, Evelyn. I wrote a very nice talk, and apologies for citing Camilla before you, but after all, she's my PhD student.
Yeah, you're very proud.
So my question was to you, the data you presented on NFL and PD. You showed that there was actually no increase in PD in contrast to others. And I showed the data unpublished data from King's, where we found the same, but we also found that it seemed still to predict cognitive worsening. So do you think that is possible? And
yes, I think that is possible. And what I showed was this review in PD patients, but this was usually, I think, at time of diagnosis, and it's on a group level. So then we don't see a lot of difference with healthy controls. But I think if you zoom into those patients who have cognitive decline, you look at longitudinal data, I think that in the group of PD with relatively elevated NFL levels, you could see this yeah, this much more cognitive decline than in the patients who don't have that. But yeah, I think interesting work and something we should look a bit deeper into. Yeah.
Thank you.
Thank you. Okay, Professor Onofri, do you have any question or for Evelyn?
Well, I enjoyed your talk, and as I guess I had the question, but about another paper that you wrote. But you are talking about stratification. What really intrigues me is the level of stratification correlation. I understand is really too poor because the cognition is rated on a very shortcut level, like the mini mental state or rather than OCA. And also I'm curious about other clinical elements which are quite important in addressing the diagnosis, like the presence of neuropsychiatric symptoms. Don't you agree that an improvement in the scales is needed in order to understand what are the correlations with the different fluid biomarkers?
Yeah, thank you for this very nice question. And I'm really curious about which paper you want to talk to me about, but you have my email address. No, I think, yes, we need some specific outcome measures for DOB. And I think we can use them. We use AD scales, we use PD scales, but they're not always fit for this type of patients. And especially also for psychiatric, and I think Dar can elaborate on this much more, but for psychiatric features, most of the time the NPI, for instance, is used, which is, I think, not a very good scale. And one of the problems with that scale is that it's based on caregiver input. And we have looked in our, there was a small scale developed here in our hospital, which was actually patient derived. And then we saw a mismatch with the MPI, and then much more hallucinations reported by patients. If we asked them without the caregiver there, then it was reported by the caregiver. So I think we should not underestimate this feature. Yeah. So I agree. We need better scales for DOB. Yeah.
Just consider how poor is the assessment still now, the assessment of fluctuations and the understanding of fluctuations. Right?
Yes. I think fluctuations are a very hard concept to grasp. Also, I think also clinicians still are struggling with that. What do we call fluctuations? I mean, everybody has ups and downs during the day. So I think that we need a better scale and also maybe a better grasp of this concept as well. Yeah.
Thank you.
Thank you. Professor Onofri, thank you very much, Evelyn.
Thank you. And have a very nice conference. I will have to leave a bit earlier, but I will stay on. Thank you.
Take care. Bye.
See you in Newcastle.
Yes, looking forward to it. So Professor Onofri, would you like to introduce the two speakers? The last two speakers. So we have Roberto is just for recorder, and Dennis will be me.
I do not know how could I introduce someone.
Sorry, Professor Bonanni, sorry to interrupt you, but maybe we have another question in the question and answers. Maria Camila Gonzalez.
Oh, yes.
Thank you.
Evelyn, are you still here?
Sorry to interrupt you.
Okay, thank you.
Yes, I'm here.
I'm sorry, Camila. Thank you, Professor Lefstra. What about the impact of ethnicity? We have seen in the past that ethnicity, more specifically Mexican Americans, have lower rates of AD positivity, but still great cognitive decline. That's interesting.
It's very interesting. And to be honest, I don't know, actually. I think that's something that's coming up now, that we look at diversity as an important factor, not only ethnicity, but also gender. How does that influence our results? And I think it should be much more aware. And so I think it's great that we have that EDLB is becoming a global effort, and we can include different race and different cohorts from all over the world to maybe tackle this problem. But I don't have the answer for you now, Camilla. Thanks for asking.
Laura, devi riaccendere, ti si è spento il microfono.
So, yes, so Professor Noffi, would you like to keep moderating the rest of the meeting?
No, I would rather like to introduce Laura like now, but because Roberto is not here, he'll send out a video recording. So actually Roberto Ceravolo is now in Pisa and he is mostly involved in Parkinson's disease. His interest shifted also on Lewy body, and he will show us his video recording of a presentation about imaging biomarkers. Unfortunately, there will be no chance to ask questions after the video recording. So he can start it directly.
Yes, we are going to show the presentation.
Okay.
Good evening. My name is Roberto Ceravolo. I am professor of neurology in Pisa, the University of Pisa, and my topic today will cover the imaging biomarkers in dementia with the Levy body. Really, the spectrum of Parkinson's dementia is a wide continuum from a pure motor Parkinson's disease to dementia with the Levy body up to Alzheimer's disease with and without extrapyramidal signs. And in the middle, Parkinson's disease with different grades and types of cognitive impairment. The evolution of diagnostic criteria for dementia with the Levy body is very interesting in this respect, because we started in 1996 with the first consensus criteria, and then in 2006, the first revision with the novelty represented by the introduction of suggestive and supportive features, including the imaging data. And these imaging values were finally confirmed in the last version of 2017, including SPECT for dopaminergic synapses, cardiac scintigraphy, and to a lesser degree, MRI, structural MRI, looking at the preservation of medial temporal lobe structures. We can visualize, as I know, dopaminergic synapses in several ways, but the most common, mainly since available for SPECT, is the investigation of presynaptic dopamine transporter density. By means of this approach, in this large multi-center study, high specificity and a good sensitivity was reported in the differentiation between dementia with the Levy body and Alzheimer's disease. Even in DLB cases without Parkinsonism, so confirming that the alteration of the nigrostriatal pathway is a molecular signature of such disease. And in Alzheimer's disease, this investigation should be always normal, even in the presence of Parkinsonism, emphasizing again the absolute value of such measure in the differentiation between Alzheimer's disease and dementia with the Levy body. But one still unsolved question is how to distinguish between dementia with the Levy bodies from Parkinson's disease with dementia. We know that the solution is an arbitrary rule, so-called one-year rule, for which if dementia onset is within the first 12 months after the onset of Parkinsonism, we can diagnose dementia with the Levy body. Otherwise, PDD Parkinson's disease with dementia. The wide overlap of such conditions is confirmed by the lack of differences on clinical grounds, cognitive, motor, as well as the positive response to levodopa. In this respect, could be imaging helpful in differentiate PDD from DLB? Several studies focused on this point, but they in total, they've provided negative results also by study perfusion as well as the metabolic profile. Also, in this study from our group, the results were discouraging. So the answer is that there are no difference in flow, metabolism, and the nigrostriatal degeneration between PDD and DLB. And the answer is that imaging is not helpful in this respect. Moreover, a recent point that we could consider is the relationship between the values of dopaminergic transporter striatal uptake and nigro cell counts. Some reports seem to indicate a linear relationship, but a more recent evidence confuted this dogma. We should, in fact, take into account that the dopaminergic transporter can be changed due to compensatory phenomena. And the main compensation is related to a downregulation of this presynaptic protein. Such compensatory mechanism is more efficient in early phases and in young onset disease with dopamine deficits. In a huge cohort of Parkinson's disease with different disease onset, we recently reported that in younger people, the impairment of dopamine transporter density is much more evident than in older ones when compared to a large control age-matched people. This is so true that older people with a typical good responder Parkinson's disease can have a DAT imaging within normal limits, as recently reported in this our case series. From this point of view, it could be useful to look at the nigra alterations more than the striatal ones. And by means of MRI with peculiar sequences, by looking at the neuromelanin content or at iron deposition, we can assess the nigra alterations present in degenerative disease with a morphological approach which does not depend on compensatory phenomena due to pharmacological changes. The possibility to assess amyloid load in vivo in the brain by imaging could raise another question. Could be the amyloid imaging helpful in this respect? I mean, in differentiation Parkinson's disease with dementia from dementia with the Levy body. By reviewing the different papers on this topic, the results are controversial. If some papers have shown a clear difference between dementia with the Levy body, in which high cerebral retention of amyloid tracers was reported, and Parkinson's disease with dementia, in which a very low retention of tracer was observed. In several other reports, a wide overlapping in amyloid accumulation was reported in comparison between DLB and PDD. However, in this systematic review to estimate the prevalence of amyloid deposition among patients with synucleinopathy and various degree of cognitive impairment, higher prevalence of positive studies was encountered among subjects with DLB as opposed to subjects with PDD. So we might suppose a situation like this for PD and PDD, situation more similar to control, and for DLB, a situation intermediate value of cortical retention in comparison with the classical amyloidopathy represented by Alzheimer's disease. At the moment, the most, I mean, the answer to the question about amyloid imaging anyway is negative. I mean, also amyloid imaging cannot distinguish between Parkinson's disease and dementia and Levy body disease, because on an individual basis, amyloid imaging cannot do this distinction. At the moment, the most reasonable view is that when amyloidopathy co-occurs with synucleinopathy, probably we have a shorter time to dementia, a shorter disease duration, and probably a worse cognitive decline, as we observed in a recent paper. The role of imaging, however, is also to let us to increase the knowledge in the physiopathology of the main clinical features of synucleinopathies by allowing us a sort of functional exploration of different nuclei. If we look at the core features of DLB and also included in the criteria for prodromal DLB, we can try to, investigate which nuclei are involved in these core features, in these specific clinical parameters. First of all, the typical fluctuation of cognitivity, I mean the fluctuating cognition. This is not clear so far. Anyway, we tried in this study to compare a cohort of Alzheimer's disease and DLB matched for age, sex, and severity of dementia, different only for the presence of fluctuation, cognitive fluctuation in DLB and not in AD. The populations were scanned by PET with FDG, and we observed that PET with FDG, the metabolic pattern by PET with FDG, showed a clear hypometabolism in the thalamus in DLB in comparison with Alzheimer's disease, well related to cognitive fluctuation. So, in some way focusing on thalamus as a probable nucleus involved in this peculiar behavior of cognitivity in dementia with the Levy body. REM behavioral disorder is another typical feature for DLB, and we know that the main role for modulation of the sleep-awake cycle and for arousal system is locus coeruleus, that is the principal source of a noradrenergic ascending pathway. It is possible to explore morphologically locus coeruleus with the neuromelanin MRI, but it is now possible to study the function of these noradrenergic synapses with a ligand for noradrenaline transporter. In a recent paper, a very good correlation between noradrenergic deficiency in vivo and severity of REM behavioral disorder was demonstrated. Finally, the hallucination, the psychosis are a core feature of dementia with the Levy body, and it is been long hypothesized that hallucinations are a clinical marker of synucleinopathies more than amyloidopathy. This hypothesis has been definitely confirmed in this recently pathologically proven large study in which a clear relationship between psychosis and Levy body load in the brain was demonstrated. We know that visual hallucinations are associated with the hypofunction in occipital areas, as is shown by several different papers by means of perfusional SPECT, metabolic PET, and MRI, structural and functional MRI. And we also know that in PD, the strongest predictor for psychosis is dementia, and more specifically, visual spatial dysfunction. The role of cholinergic ascending projection is a recent focus of attention, since it is possible now to study the structures of nucleus basalis of Maynard. And several papers, recent papers, have emphasized how the volume of nucleus basalis of Maynard is a strong predictor for dementia in PD not yet demented. But small volume of nucleus basalis of Maynard in Parkinson's disease at baseline can also predict the occurrence of psychosis. The interesting point is that when the nucleus basalis of Maynard was electrically stimulated by deep brain stimulation, more than a beneficial effect on dementia, a positive effect on psychosis was reported. For the phase 1 clinical trial, looking at the effect of nucleus basalis of Maynard stimulation in dementia of Levy body was a failure. Anyway, I mean, only recently we have understood the complex relationship between nucleus basalis of Maynard and cholinergic dysfunction and psychosis in synucleinopathies, because a direct connection from nucleus basalis of Maynard to temporal and occipital regions were recently demonstrated, so confirming the main role of this nucleus in modulating one of the typical core features of dementia with the Levy bodies, such as hallucination. So, I mean, in conclusion, I mean, as a take-home message, a PET SPECT of dopaminergic synapses can distinguish synucleinopathies from Alzheimer's disease, but cannot distinguish dementia with the Levy body, Parkinson's disease with dementia. For true, this approach should be considered as the proof of nigrous striatal degeneration, but we must be careful about compensatory mechanism age-related. In this case, in this respect, probably we should think about nigral alteration, to look at the nigral alteration by MRI, neuromelanin, or iron-sensitive sequences. The metabolic or perfusional signature cannot identify on an individual basis dementia with the Levy body. In vivo investigation of amyloid cerebral load could indicate a major contribution of amyloid in dementia with the Levy body than in Parkinson's disease with dementia. However, it is not useful on an individual basis for diagnostic confirmation. Even in the comparison between dementia with the Levy body and Alzheimer's disease, the absolute value is scarce and is not reliable, because we can find amyloid load also in dementia with the Levy body. In vivo assessment of noradrenergic terminals is able to show their involvement in REM behavioral disorder. And finally, and recently, the assessment of nucleus basalis of Maynard and its cholinergic projection reveal their role in modulating psychosis, visual hallucination in dementia with Levy body. And with this, I conclude my lecture, and I thank you, all of you, for your attention. Good evening. My name is Roberto Ceravolo. I am professor of neurology.
Okay, thank you, Professor Onofre.
Well, actually, we have agreed that we are not going to allow questions. And so actually, my final last contribution would be to present Laura Bonanni, but I guess everybody knows her here in this circumstance. So Laura has worked with me for more than 20 years. She
don't say that.
She started with basic neurophysiology, and then she moved to clinical neurophysiology. She developed how can I say, the punctualization of the application of EEG to Lewy body dementia, and then actually she's working mostly the last lectures she's done were on artificial intelligence, but she'll not deal with it today since she'll have to talk about neurophysiological biomarkers, and these are not part of the artificial intelligence, but will be. So Laura, please give us your last lecture and we conclude.
Thank you, Professor Onofre. I will ask Roberta to upload my presentation. Okay, can I go? Okay, let's see if it works. Okay, so first I would like to thank the attendants to stay so long until late to listen to my story, which is actually the story that I would like to tell is at the basis of why we should keep looking at EEG rhythms in DLB. Rhythmicities in the EEG signal are thought or believed to be caused by neuronal synchronization from an extensive inhibitory processes within a specific network, which is the thalamocortical system, or from negative feedback among excitatory and inhibitory neurons. So it's very complex as a network, or both, depending upon the frequency of interest that we are looking at. By its definition, every rhythmic signal is periodic and is relatively easy to analyze given the regularity of this feature. So it's very like a basis of what it's easy to look at by recording an EEG on our scalp. The thalamocortical rhythms, and specifically the thalamocortical neurons, have the firing properties and ionic conductances, which are very specific and peculiar. They respond to input from sensory pathways of the cortex by discharging in two specific modes, a tonic burst and a rhythmic burst. And this discharge affects the pattern of the EEG that we can record on our cortex. Tonic sphingospheric firing occurs during wakefulness or REM sleep, while rhythmic burst firing occurs during non-REM sleep. So every stage of consciousness has a specific activation of the thalamocortical neurons. The two patterns of discharge depend on multiple states, including a resting membranal potential and activation of calcium channels, T-type calcium channels, which are postsynaptic, and the inputs, which can be excitatory from cortex or inhibitory from reticular nuclei, ascending modulatory input from brainstem cholinergic and monoaminergic nuclei. So the thalamus in this system drives network oscillation through the cortical projections. So basically the thalamus plays a role in orchestrating, like it was a pacemaker, the change in the discharge pattern of cortical neurons that underlies the EEG difference between different states of consciousness, and specifically from wakefulness and non-REM sleep. So this is because the thalamic neurons, like the cortical pyramidal neurons, have intrinsic membrane properties that can cause the discharge pattern to change as a function of the level of depolarization of the cell. And basically, we can say that what happens in the thalamus can be recorded in the cortex. They are very, very strictly connected. So basically, at the same frequency that is arising in the thalamus has a counterpart in the cortex. So that's why we say that the thalamus make like a pace. It's a pacemaker for the cortex. And I would like to talk about the story of the thalamocortical dysrhythmia model. What is the thalamocortical dysrhythmia? This theory were based on old studies performed by Steriade in stereo EEG recordings performed during stereotactic surgery, which show the low threshold bursting activity with a theta rhythmicity in medial thalamic nuclei was present in patients with Parkinson's disease, and also afterwards we have seen in patients with Lewy body dementia. So cortical EEG recordings showed a theta activity which was coherent with the thalamic slow theta rhythms. So basically in these patients, in PD patients in DLB, there were a theta rhythms which was recorded in the cortex, which was very specific of the thalamic activity. So these papers, this first paper, original paper, suggested that in several disorders, including LBD, the thalamocortical interactions may enter a dysrhythmic state, which can be focal, lobar, or widespread, characterized by coherence to slow theta rhythms between 4 and 8 Hz, which are slower than the alpha activity that we always have in resting state in physiological condition. This replaces also the tonic gamma band activity linked to sensory motor and cognitive functions and tasks, and the alpha activity during waking rest state. The thalamocortical dysrhythmia model hypothesized that a portion of the thalamocortical system is kind of trapped in single-like theta activity, whereas other parts of the brain remain in the waking state, with gamma band activation at the edge of the dysrhythmia, locked brain portions, in a condition which is very similar to dissociated states of parasomnia. More specifically, the basic question at hand while we still think about the thalamocortical dysrhythmia: what happens if a set of neurons in the thalamus displays low rhythmicity and otherwise awakened brain state? This is a very old work by Mahowal and Schenk, who hypothesized for state of dissociation between cortex and thalamus. So basically the possibilities are represented by dissociation from prevailing wakefulness due to intrusion of feature of other stages of wakefulness or consciousness into the awakened brain. It is called within mind dissociation. In this state, hypnagogic or hypnopompic hallucinations occur, and complex nocturnal hallucination can appear, in which dream mentation happens during the transition from sleep to wakefulness and vice versa. A second possibility is the dissociation from non-REM sleep due to intrusion of features of other stages into ongoing non-REM sleep. The third state is called mind-body dissociation. It's represented by dissociation from non-REM sleep. Can you hear me? Due to intrusion of features of other stages, for intrusion of feature of other stages into the REM sleep, the mind-body dissociation. In this condition, the mind is asleep. We have a REM dream mentation while the body is awake, because the spinal motor neurons, which should be inhibited, are still excitable. And the first possibility is the status dissociatus, where motor agitation occurs with enacting of dreams on areas, continuous or semi-continuous movement during dream, impaired level of vigilance during daylight, with fluctuation in attention, and sometimes confabulation and mental confusions. We probably, most of you already have seen this slide, which is, I'm very fond of this slide. Why? Because if we think about what are these 4 dissociation states, basically we're talking about visual hallucinations, parasomnias, REM sleep behavior disorder, delirium, and cognitive fluctuations, the very core features for the diagnosis of DLB. So our hypothesis is that probably at the basis of EEG alteration that we will show in DLB, there is a thalamocortical dysrhythmia. So we have a story about our EEG studies. What is the evidence of thalamocortical dysrhythmia in DLB? The EEG that we record in DLB patients is characterized by the appearance of a fast theta that we called in our original paper pre-alpha activity during wakefulness, that appears, for example, in this representation, which is a compressed spectral array representation, as the appearance of a peak of frequency slower than the alpha activity. Here we have a peak of frequency, which is actually in the theta band, fast theta band, or 6.5 in this case, or intermingle with the even slower activity at 3 years. So the change between pre-alpha activity and the delta activity without any presence of alpha activity. So based on these first results, the presence of this thalamic rhythm in EEG of patients with DLB actually predicts the occurrence of cognitive decline, tracks the evolution of mild cognitive impairment in DLB, and is correlated with the severity of cognitive fluctuations in DLB. This is just the introduction of what I would like to talk about today. So the pre-alpha activity appears initially as a pseudoperiodic inscription of the normal resting state background alpha activity predominantly in the frontal regions, and becomes with time diffusely present on all scalp derivations recording cortical activity. This pattern is rhythmic, as typical of the thalamocortical spindling activity, as we have seen in the first slide. And in patients with severe dementia, this pre-alpha becomes less clearly distinguishable and is concealed by the appearance of slower theta-delta rhythmic activities, which are due to cortical disconnections. Well, for the first time, based on our original studies, basically EEG features were accepted as a biomarker of a neurodegenerative condition. It seemed to be very specific to DLB as compared to Alzheimer's disease. So Alzheimer's disease don't seem to have these thalamocortical dysrhythmia features. And the presence of the pre-alpha rhythm, the thalamocortical dysrhythmia typical rhythm, seemed to be very robust among different studies coming from different laboratories. And these are all the studies that were performed in those years from the 2008 and last couple of years. So a lot of studies around like 15 years, basically, from different laboratories, all showing the presence in DLB as compared to Alzheimer's disease of a pre-alpha cortical activity. So this is the premise of what I would like to talk. So in the diagnosis of Lewy body dementia, now we have as a supportive biomarker the presence of this EEG alteration. And actually these EEG alterations were present also in the prodromal stage of the disease, and specifically in the MCI type of DLB, prodromal DLB. These are a study from our laboratory, and this is a study from Evelyn laboratory, where we both show the presence of a pre-alpha frequency with a pattern of thalamocortical dysrhythmia in MCI DLB patients as compared to non-converter and to MCI Alzheimer's disease patients, and this was very, very consistent between the two laboratories. In this graph, we can see here that the blue line, the violet line, is represented by the presence of slower activity. The peak is around Hertz. It's a pre-alpha activity present in the prodromal MCI DLB, while the Alzheimer's disease, prodromal Alzheimer's disease, with MCI, basically, had an alpha activity, which was much more prevalent as compared to DLB. So very strong. But prodromal DLB is not only MCI. Actually, we know that we have different clusters, as Doug pointed out before, and among them, there is also a delirium onset DLB. And if we remember the slide about the difference, the dissociation state, the 4 dissociation state that they presented was the status dissociatus, which was very similar as clinical feature to a delirium. So our hypothesis here today, and I'm presenting the preliminary data of this study, is if delirium is one of the dissociation state presented by Mahowal and Schenk as a part of the thalamocortical dysrhythmia, And as DLB as a feature in the cortical activity typical of thalamocortical dysrhythmia, maybe we have found the basis of the delirium type prodromal DLB. So what we did, what have we done in our laboratory last year? The aim was this one. So since delirium resembles the status dissociatus of TCD, thalamocortical dysrhythmia model, and since delirium represents a possible presentation of prodromal DLB, we aim to evaluate whether the EEG study can support the hypothesis that thalamocortical dysrhythmia represents a pathophysiological mechanism underlying the onset of delirium, to evaluate whether a group of cognitively preserved patients, so non-demented patients who develop an episode of delirium during hospitalization, have any changes in their EEG typical of thalamocortical dysrhythmia, and to identify an EEG pattern common to delirium and DLB, to demonstrate the possible role of thalamocortical dysrhythmia in the pathogenesis of both these clinical syndromes, and specifically in the delirium onset DLB cluster. So this is the very first study. These are the materials and methods. We recruited 91 patients admitted to our neurology clinic between 2020 and 2021. We had a lot of people with delirium. Some of those were patients with a stroke, we will see. And the presence of delirium was checked by the 480 test and was based on the diagnostic criteria of the DSM-5. These are the inclusion-exclusion criteria, basically absence of cognitive decline. They had to have a brain neuroimaging examination and informed consent. Among the exclusion criteria, MCI or dementia, previous psychiatric disorder, history of alcohol abuse, pre-existence of EEG abnormalities, and also the presence of thalamic lesions or atrophy on brain CT or MRI. So we did in a EEG examination, amnestic data, and lots of examination about comorbidities and cause of hospitalization, basically. These are the pattern that we try to analyze in our EEG study. Basically, we can say that a pattern of one, which is the presence of a normal activity, alpha activity in the occipital derivation, was deemed as physiological, while the other patterns, which were originally described in our first words, are basically the presence of the pre-alpha activity. We also these patients. So for 6 to 12 months, the patients were asked after the discharge from hospital to go to clinical instrumental evaluation to investigate the possible onset of a cognitive decline, and also to see which kind of cognitive decline they developed. So again, they performed a EEG, a control EEG, FDG PET, and caregiver interview to see for the presence of or appearance of RBD, cognitive fluctuation or visual hallucination, possibly. And as you can see here, very complex neuropsychological evaluation to look for early and very mild sign of cognitive decline. I would like in this case to thank especially Claudia Carrarini, who carried out the study and the design of the follow-up and specifically the neuropsychological evaluation. So these are the results. Unfortunately, this is a very preliminary study because many patients were excluded from the 21 admitted at the beginning because many of them had actually dementia, and some of them couldn't perform type of delirium, which actually led us to have an impossible recording or a very full of artifacts. So the final groups were represented by 14 patients with delirium and 13 patients without delirium. They did not develop delirium during hospitalization. These are the demographic and clinical characteristics that were well matched as age and demographic characteristic and predisposing factors. There was no difference between non-delirium and delirium group. What we found, it was just a very little, maybe a difference in age, but basically they were very, very similar. So there were no difference predisposing factors, no difference as for reason of the admission, and the most frequent reason for admission was acute cerebral ischemia. The group of delirium had an onset after 2.6 days, so it's very early after the hospitalization, and the mixed delirium type was the most frequent subtype found in our patients. 2 patients deceased in the delirium group. How was the EEG in these 2 groups of patients? Basically, the delirium group had in the very high majority, basically in the 92.9%, 93%, a pathological pattern very typical of DLB with the presence of the pre-alpha activity, while the delirium basically had mostly a normal EEG pattern. In the case of non-delirium group, we found in 5, so 40%, basically 40% of the patient, a pathological pattern. So it's very striking difference in this very first preliminary study. So the nominal frequency was significantly lower in the delirium group because we had the pre-alpha activity. The dominant frequency variability, which is the presence, which signs the presence of 2 peaks of activities in the delirium group as compared to non-delirium group, which actually showed a very stable dominant frequency around the epochs in the alpha band. And the pathological pattern, so the CSA pattern that we found before, was highly present in delirium group as compared to non-delirium group. So what we can say from this preliminary result, we also performed a logistic regression models, and we found a significant association between delirium and the presence of both a pathological CSA pattern, EEG pattern, and a dominant frequency slower than alpha, the pre-alpha dominant frequency. In the follow-up, only 5 patients in this very short follow-up, 6 months, belonging to delirium group underwent follow-up visits. Some of them did not come, and as you can have seen before, they were the years of the pandemic, so basically patients did not come very easily at follow-up. All patients were evaluated and presented a clinical picture of dementia. These 5 patients, the 5 patients that came, they were demented after 6 to 12 months after admission. And the EEG at the follow-up basically showed again the presence of a pre-alpha dominant frequency. So it's very similar to what we found at the beginning of admission of the study. Only 1 patient underwent so far a PET, FDG PET, and this patient had a moderate posterior hypometabolism with a singular Italian sign, again, very specific of DLB. So these very preliminary results seem to be at least promising. So what we can discuss here is that the different variables analyzed, such the EEG pattern, the dominant frequency, showed a significant and distinct characteristic in patients with delirium compared to those without delirium. And compared to non-delirium group, the patients with delirium had mainly a dominant frequency in the pre-alpha band, typical of thalamocortical dysrhythmia. So maybe the thalamocortical dysrhythmia determines the onset of a non-ordinary states of consciousness, and maybe also to delirium. Maybe it's the basis of the delirium. The symptoms observed in delirium recalls the clinical characteristic observed in DLB, in particular cognitive fluctuations. Delirium is indeed considered, as we have seen, a prodromal manifestation of DLB. So the appearance of a pre-alpha activity in EEG recordings is considered a supportive biomarker for the diagnosis of DLB, and the presence of pathological EEG patterns in the delirium group suggests a possible involvement of thalamocortical dysrhythmia and the pathophysiology also of delirium. Considering delirium as a prodromal symptom of DLB, the observed EEG could document a possible onset of dementia, particularly of the DLB type. And there is currently also a lack of a validated diagnostic criteria from the delirium cluster onset of prodromal DLB. What are the strengths and limitations of the work? We probably have found a possible involvement of thalamocortical dysrhythmia and the pathophysiology of delirium. So maybe EEG is potentially useful as a diagnostic tool to identify patients in the prodromal phase of the disease, even in the delirium cluster. This can lead to possible pharmacological intervention on the neurotransmitter involved in thalamocortical dysrhythmia, the cholinergic system, also in patients with delirium. of course, the limitation of the small sample size, the single center study, and we need to expand the longitudinal study. But we are working on it, and we actually performing a multi-center study. We were able to recruit a high number of patients now, 65 patients with delirium and 41 patients without delirium, thanks to the efforts of all these centers involved in the multi-center study. But today, I'm not going to present the results, because if you want to see more of this story, you need to come to Newcastle and to hear the all multi-center story results. And finally, I would like to also thank all the attendants, all the speakers that came to our webinar, and again, the Center Formazione a Distanza, Comunicazione e Congressi. Thank you very much. I see that I have some questions in the question and answer section. I can read it myself. Claudio, thanks to Professor Bonanni from this innovative research on the neurophysiological oscillatory thalamocortical at resting state, fast theta pre-alpha rhythms underpinnings of delirium as an early symptom of LV disease. Can you get into more details about the topography of those EEG rhythms, delirium patients, and issue to frequencies? What about the level of vigilance of them during the EEG recordings? It's very interesting. It's a technical question. So basically, the presence of pre-alpha frequency, and we divided the scalp in three main regions: a posterior, including parieto-occipital derivations, temporal derivation, and anterior, which included basically frontal derivations. We didn't find any great difference between the three regions, beside the fact that in frontal derivation we found more artifacts. And in Newcastle we'll present more in details the data. We had to exclude some of the patients because we found too many artifacts in the frontal derivations. About the vigilance of them, it's very hard to say, because most of the patients had mixed delirium. We decided to record the patients while they were not agitated, of course, to avoid the presence of movement artifacts. But it's very hard to assess the vigilance while you have a patient admitted to the hospital. We usually have, when we do a resting state, basically the neurophysiology technician asks the patients every time, each time to assess if he's sleeping or he's awake. Alberto, hi Alberto, thank you for coming. Thank you, Professor Alberto Jaramillo Jimenez is a collaborator of Professor Arslan. He is a great neurophysiologist. He's performing brilliant studies on EEG in many neurodegenerative diseases. Thank you, Professor Bonanni, brilliant presentation. Thank you, amazing ongoing study. As researchers, we are considering both the slowing down represented as a posterior dominant frequency at pre-alpha, as well as the variability of the dominant frequency for DLD criteria. Do you think these features alone are good markers for treatment studies? Should we look for a composite EEG matrix or go for multi-domain studies mixing CSF, genetic, and EEG markers? Absolutely, yes. We need to go for a combined biomarker. And as Evelyn pointed out before, actually NFL, for example, are correlated, as she said, with the presence of a synuclein and of specific features of atrophy in the brain. So maybe combining also EEG with these markers can be definitely helpful.
There's another question.
Oh, there's a question from Doug. Grazie Laura, perfetto. Can EEG help showing treatment response or predict response? Definitely yes. We have several studies. And Professor Onofri actually performed several studies in the past looking for the effects of cholinergic medications, on EEG activity and the dominant frequency variability. Definitely, we have room for try to do specific pharmacological studies, EEG pharmacological studies.
Thank you, Laura. Thank you for your presentation. And it's hard to wait till Newcastle to have the final results of the delirium study, but we have to wait
Thanks.
Exciting.
Thank you. Okay, if there is any other question, I will ask Professor Onofre to wrap up and I will just thank everybody from my side. Microfono.
Is it working? I would like to thank particularly Doug Arslan, who had the strength to stay until the last lecture. And all the other audience. And I'm very glad that also this year we could complete this webinar, which we hope will be the last one, since from next year on, we hope in a better organization and presence with all the people who's working on the field. So thank you to everybody, to the few which are still staying, and we'll meet probably in Newcastle, or if not, in the other next meetings. Bye. Bye.
Thank you. Bye-bye.
Bye, ciao.
Have a nice evening.
Thank you, everybody,
Bye.
for being
Bye.
here. Bye-bye, everybody.
Bye.